the machinery responsible for the shape-shifting of Zika, along with a potential vulnerability. The vital enzyme of Zika virus carries out various functions, but any disruption in its operation could abruptly halt its activities. Greetings, everyone. In today's video, we will be discussing a significant breakthrough made by researchers at Sanford Burnham Prebys. They have successfully identified a potential vulnerability in the Zika virus that could be targeted with medication to halt its replication. The team's findings revealed that the NS2BNS3 enzyme of the virus possesses the ability to break down proteins and separate its double-stranded RNA into single strands. This enzyme's functionality is dependent on its shape and can transition between open and super-open conformations, allowing it to grasp and release a single RNA strand. By developing a drug that inhibits Zika's conformational changes, we may have a promising method to prevent the virus from multiplying. In the realm of viruses, genetic material is scarce, and proteins are in short supply, requiring every component to exert maximum effort. A prime illustration of this is the Zika virus, which only generates 10 proteins. Recently, a team of researchers from Sanford Burnham Prebys conducted a study published in the journal PLOS Pathogens, unveiling the virus's remarkable efficiency and potentially pinpointing a vulnerability that could be targeted for therapeutic purposes. During the investigation, the team of researchers demonstrated the versatility of Zika's enzyme, known as NS2BNS3, which serves two crucial purposes: protein fragmentation as a protease and the separation of its own double-stranded RNA into individual strands as a helicase. According to the senior author of the paper, Dr. Alexey Terskik, an associate professor at Sanford Burnham Prebys, the enzyme complex of the Zika virus exhibits different functions depending on its shape. In its closed conformation, it acts as a typical protease. However, it undergoes a cycle of transitioning between open and super-open conformations, enabling it to capture and subsequently release a single strand of RNA. These particular functions are vital for the replication of the virus. Belonging to the family of perilous pathogens known as flaviviruses, Zika is an RNA virus. Other members of this family include West Nile, dengue fever, yellow fever, Japanese encephalitis, and various others. Mosquitoes serve as carriers of this virus, which has the ability to invade uterine and placental cells, posing a significant threat to expectant mothers. Upon entering host cells, Zika manipulates their genetic makeup to facilitate its own replication. The potential benefits of comprehending the molecular intricacies of Zika are substantial, particularly in identifying a target for therapeutic intervention. Developing drugs that specifically target the enzyme domains responsible for protease or helicase functions is challenging due to the presence of similar molecules in human cells. Nevertheless, a promising approach would be to design a drug that inhibits Zika's conformational changes. By preventing the complex from undergoing shapeshifting, its crucial functions would be rendered ineffective, thereby halting the production of new Zika particles. For quite some time, scientists have been aware that the crucial enzyme of the Zika virus consisted of two components, NS2BNS3-PRO and NS3-HEL. NS2BNS3-PRO is responsible for executing protease functions, which involves cleaving lengthy polypeptides into Zika proteins. However, the newfound understanding of NS2BNS3-PRO's capacity to bind single-stranded RNA and facilitate the separation of double-stranded RNA during the replication of the virus has only emerged in recent times. The researchers in this study utilized up-to-date crystal structures and employed protein biochemistry, fluorescence polarization, and computer modeling to analyze the life cycle of NS2BNS3-PRO. NS3-PRO is joined to NS3-HEL, the helicase, by a brief amino acid linker and is activated when the complex assumes its closed conformation, resembling a closed accordion. RNA binding occurs when the complex is in its open state, while the complex must transition into the super-open conformation in order to release RNA. The movement and alterations in shape occur due to the activity of NS3-HEL, which propels the linker forward and eventually pulls NS3-PRO, resulting in the release of RNA. NS3-PRO is firmly attached to the interior of the host cell's endoplasmic reticulum. a vital organelle responsible for guiding cellular proteins to their designated locations. NS3-Pro, in its closed form, plays a crucial role in cleaving the Zika polypeptide, contributing to the production of all viral proteins, and is supported by NS2B. Conveniently positioned on the opposite end of the linker, NS3-Hel effectively divides the double-stranded RNA of Zika and proceeds to pass one of the strands to NS3-Pro. This transfer is facilitated by the presence of positively charged forks on NS3-Pro, which securely grasp onto the negatively charged RNA. According to Turskik, there exists a delightful alignment of positive charges that attracts RNA, causing it to effortlessly navigate along this path. Subsequently, the complex undergoes a transformation into a closed conformation, resulting in the release of the RNA. When NS3-Hel extends its reach to seize the double-stranded RNA, it brings along the entire complex. However, due to the anchoring of NS3-Pro in the ER membrane and the limited extension capacity of the linker, the complex abruptly shifts into the super-open conformation, resulting in the release of RNA. Subsequently, the complex returns to its open conformation, poised for a fresh cycle. In the reverse inchworm process, NS3-Pro transforms into a protease when it identifies a viral polypeptide to cleave, causing the complex to adopt a closed conformation. This phenomenon, as described by the authors, is akin to inchworm-like movements in reverse, with the protease, referred to as the jaws, trailing behind as it grasps and releases the single-stranded RNA. Not only does this comprehensive comprehension offer a potential avenue for treating Zika, but it also has the potential to be utilized in the study of other flaviviruses that possess comparable molecular mechanisms. According to Turskik, the NS2-BNS3-Pro complex can be identified in various flaviviruses. This discovery has the potential to introduce an entirely novel category of drug targets that can be utilized against multiple viruses. To support our channel's growth and ensure broader awareness, kindly hit the like and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.